Welcome to Week 8 of Applied Immunology. Following the Week 6 and 7 lectures on experimental and diagnostic tools, starting this week we are going to enter the final section of the course where we start to learn more about immunological diseases, as well as different types of therapies that are based on manipulating the immune system. In Week 8, we will cover one of the most important applications of immunological therapies, which are vaccines. But I want to first start off with today's Lecture 8A, which will tie together most topics we've learned about so far by summarizing the sequence of events that occur during a comprehensive immune response to infection. We will begin by revisiting the general goals and timing of a successful immune response, then discuss an example of bacterial infection, and examine how innate and adaptive immune cells coordinate their activities together. Lastly, we will cover some mechanisms involved in the resolution of inflammation following infection clearance. Today's lecture will recap a lot of topics that we already covered in weeks 1 through 5, so there's not going to be a ton of new information. But I think that this will be a helpful lecture in terms of revisiting some of this info in a way that really emphasizes how immune cells and pathways coordinate their functions to promote host defense from pathogens, rather than only working as a single pathway or cell type in isolation. I also think that reviewing these main points should be a useful refresher before we start to learn more about immune-based therapies and diseases throughout the remainder of this course. We first saw this schematic back in week 1 when we introduced the kinetics or timing of an immune response over the course of a typical infection with something like a species of bacteria shown here in red. This can be separated into 3 phases over time along the x-axis. So we have infection and inflammation in blue, resolution in purple, and immune memory in green. We've established that this first phase of the immune response can be separated into innate immunity, which occurs immediately and is represented by the gray curve here. And we also know that innate immunity can stimulate the T and B cells of the adaptive immune system, which is slower to respond at first as represented by the black curve, but is powerful in terms of mediating antigen-specific pathogen clearance. This first phase of the immune response to infection in the blue portion of this figure corresponds to the first 3 goals of a successful immune response, which involve recognizing pathogens, engaging activation programs needed to respond to that infection, and then deploying effector mechanisms that kill the pathogen. We've learned a lot of details about the specific cell types and signaling pathways that accomplish these 3 goals, both for innate and adaptive immune responses. And we will revisit them in the next few slides as we go over an example of infection. Let's first consider a case where bacterial infection is introduced to the skin following a puncture wound. We know that epithelial surfaces such as the skin possess a variety of anatomic barriers to infection, ranging from mechanical exclusion of pathogens to chemicals like antimicrobial peptides such as defensins, as well as microbiological competition that's provided by commensal microbes. These anatomic barriers can be compromised if there's damage to the skin epithelium. The example we will use today is a puncture wound with something like a spike from this cactus that's shown here in the middle. I'm being pretty specific here because this is a picture of a cactus on my patio that actually messed up my hand pretty badly a few months ago. So I got a spike buried in my finger, it got infected, and I eventually had to have minor surgery to remove it. I'm still kind of mad about this because the whole ordeal ended up costing me a lot of money and insurance copays and wasn't the most fun use of my PTO. So I'm going to go ahead and be petty and use this cactus as an example of epithelial barrier damage and resulting infection. Now, once the skin epithelium is broken by a puncture wound like a cactus spike, bacteria can bypass these anatomic barriers to infection and access the vulnerable tissue that's located underneath the epithelium. This establishes a focus of infection locally in the skin, which needs to then be eliminated by the immune system. In this example, let's consider a bacterium that can be found extracellularly as well as intracellularly. So there are a few bacterial species whose replication involves both extra and intracellular stages. We can start by filling in some detail about which innate immune cells and signaling pathways are involved in this first early phase of infection and inflammation. The first goal is to recognize invading bacteria in the wound, which can be done by innate immune cells such as macrophages and dendritic cells shown here, as well as a variety of other cell types. The bacteria can be recognized through pattern recognition receptors, or PRRs. and we've learned that these include TLRs on the plasma membrane, as well as cytosolic NLRs, which could respond to intracellular bacteria. We've also learned about things like RLRs and C-gas sting sensing, which are more relevant for a viral infection, which is why I'm kind of leaving them out of this example. The second goal is to engage activation programs in response to pathogen detection, and we've learned about a lot of these in our lectures on innate immunity. These include inflammasome assembly, complement activation, and inflammatory gene expression in innate immune cells. In this example of a bacterial infection, these transcriptional programs are most likely to be induced by the transcription factor NF-kappa B, but we know that during viral infections you can also get activation of IRF transcription factors. NF-kappa B activation can upregulate many transcriptional targets that are important in this early response phase, including inflammatory cytokines such as IL-12 or TNF-alpha, as well as chemokines like CXCL9. Inflammatory gene transcription can also be associated with enhancing the cellular activation of professional APCs such as dendritic cells, and this sort of increases or enhances their ability to process and present antigen. The effects of these activation programs in innate immune cells lead to the execution of effector functions, which again is the third goal of the immune response. We've learned about a bunch of these, including several mechanisms that act locally to eliminate pathogens at the site of infection. These include programmed cell death, such as pyroptosis, which is accompanied by IL-1 family cytokine secretion. Complement activation leads to the opsonization and phagocytosis of bacteria by professional phagocytes, like macrophages or neutrophils. We know that these cells can then kill internalized pathogens using things like ROS and antimicrobial peptides. Remember that complement signaling can also promote bacterial lysis through MAC formation. There are also several mechanisms that can link innate immune activation to the activation of other cell types. This includes production of inflammatory cytokines that can activate the vascular endothelium, and this leads to vessel permeabilization, fluid edema, and expression of ligands that promote immune cell migration and diapedesis into infected tissues. Cytokines can also activate neighboring immune cells, such as classical innate lymphoid cells, or ILCs, which in this particular case of bacterial infection would likely be ILC1s or ILC3s. Chemokines produced by innate immune cells serve as migration cues, and so these can recruit additional leukocytes to the infected tissue. And this includes things like circulating innate immune cells like neutrophils or monocytes, as well as adaptive lymphocytes such as T and B cells. Lastly, some mechanisms are relevant for stimulating adaptive immunity. The first is the activation of professional APCs, which can stimulate adaptive T cell responses through immune priming. The second is the drainage of soluble antigens to secondary lymphoid organs, which is important for stimulating B cell responses. Both of these outcomes are extremely important in case local mechanisms of innate immunity are insufficient to clear the infection. And in this case, you would need the activation and recruitment of adaptive lymphocytes to help combat this skin infection. Adaptive immunity can be stimulated in secondary lymphoid organs, such as the tissue-draining lymph nodes, in two different ways. One is the drainage of soluble antigens through the lymphatics, which we learned about back in week 1. Lymphatic circulation is important for washing afferent lymph containing bacterial antigens over the B cell zones of the lymph node. Again, because the BCR binds to soluble antigens, this is important for triggering B cell activation, which leads to the formation of a primary focus as well as germinal centers and resulting antibody production. The second arm of adaptive immunity that can be stimulated in the skin-draining lymph node are T cells. This is accomplished through migration of activated APCs such as dendritic cells to the lymph node, where they can then present bacterial antigen to stimulate naive helper CD4 T cells, as well as cytotoxic CD8 T cells. Again, this process is often referred to as adaptive immune priming. Since immune priming requires the migration of activated APCs, such as dendritic cells, to the lymph node, I want to emphasize the important role that chemokine gradients play in orchestrating a comprehensive immune response. We've seen this figure back in week 4, which highlights that DCs activated by PAMPs in peripheral tissues such as the skin can upregulate a number of markers like co-stimulatory molecules as well as the chemokine receptor CCR7. CCR7 binds to chemokines named CCL19 and CCL21, which are expressed in the T cell zone of lymph nodes. So, once the activated dendritic cell drains out of the skin and enters lymphatic vessels, CCR7 expression is an important step that helps guide the DC to migrate towards the CCL19 and CCL21 gradient that has been established within lymph nodes. The antigen-loaded DC arrives through the afferent vessel at the top of the lymph node, and then migrates deeper into the T cell region of the lymph node that's shown in this diagram in blue. Once in the T cell zone, the organized architecture of the lymph node allows the DC to kind of crawl around and sample T cells in a process called browsing. And the dendritic cell can do this until it finds a T cell with a cognate TCR that recognizes its presented peptide MHC complex. This interaction then triggers T cell activation and the acquisition of effector functions. So we can think of this process as sort of achieving the goals of pathogen recognition and cellular activation for T cells. In this case, where we have an infection with a bacterium that has both extracellular and intracellular phases of its life cycle, T cell activation would likely be linked to the activation of CD8 positive CTL responses, as well as the differentiation of a few different helper CD4 subsets. Again, these CD4 subsets are based on the signal 3 cytokine that's produced by the APC during T cell activation. So in this specific example, we would likely see the differentiation of Th1s to combat the intracellular phase of bacterial infection, Th17s to combat the extracellular phase of infection, and T follicular helper cells to support B cell responses. Similarly to how CCR7 guides activated DC migration to the lymph nodes, once T cells are activated, their migration is also determined by chemokines. Activated TFH cells will downregulate expression of CCR7 and instead express a new receptor called CXCR5, and this recognizes the chemokine CXCL13 that's produced in B cell follicles. This means that activated T cells will leave the T cell zone, follow the CXCL13 chemokine gradient, and then migrate to the border of the B cell follicle. Here, the T cells can meet up with activated B cells that express CCR7 and have migrated to this interfollicular zone. So the result is that these two cell types can then interact with each other, and so the T cell can provide T cell help required for B cell isotype class switching as well as other processes. Chemokine receptor expression is also important for effector T cells to be able to egress or leave the lymph node and migrate to the site of infection. We learned about one of these chemotactic cues back in week 4, which is S1PR1 expression on activated T cells that allow them to follow a gradient towards S1P that's found in the efferent lymph, and this allows the T cells to exit the lymph node. Once effector T cells have left the lymph node, they enter circulation and home to the site of infection by following additional chemotactic cues. From our example of a skin infection, the endothelium that lines blood vessels in the skin expresses a chemokine called CCL17, as well as a molecule called E-selectin, and these bind to CCR4 and CLA expressed on T cells, respectively. This allows the effector T cells to exit circulation and enter the infected tissue through this process of diapedesis. Once in the tissue, these T cells can then migrate to the epithelium by following a gradient of CCL27 expressed by keratinocytes, and this binds to CCR10 that's expressed by activated effector T cells. All of these chemotactic cues function by making sure that activated T cells can physically move to the places where they are needed to respond to an infection, whether this is migrating to the T cell zone to support the B cell germinal center response, or whether this means leaving the lymph node and migrating into infected tissues where they can exert effector functions. There are many different chemokine and receptor pairings, which differ with respect to the cell types that they are expressed by, as well as the specific organs where they are located. And these really play a critical role in the spatial organization of an immune response to infection. So, although this example outlines specific chemokines that bring effector T cells to the skin, keep in mind that these signals may be different depending on the specific organ or tissue where an infection takes place. Once T cells and B cells are activated in the skin-draining lymph node over the course of several days, they can migrate to the infected tissue and exert their effector functions. This schematic from the book highlights how innate and adaptive responses are coordinated both across different physical locations, or spatially, as well as over the course of different time frames, or temporally. So you might see this referred to as the spatiotemporal coordination of a comprehensive immune response. The third goal of a successful response is to deploy effector functions, which we already covered for some types of innate immune cells earlier on in this lecture. For adaptive immune cells, the effector functions of B cells are primarily accomplished through production of soluble antibodies, which can enter circulation and be deposited in the infected skin. For this case of bacterial infection, these antibodies might consist of early stage IgM and then later stage IgG or IgA isotypes, and their most relevant functions in this particular example would be bacterial neutralization, opsonization for phagocytosis, and complement activation. The primary effector functions of T cells require their migration into tissues, which, as we just learned, is heavily dependent on chemokine gradients. Let's next learn a little bit more about some of the effector functions of Th1 cells for this example of bacterial infection, since we've established that this specific bacterium exhibits an intracellular phase of its infection cycle. Th1s are a good example to use when discussing a comprehensive immune response because they produce a lot of different effector molecules that can promote defense from intracellular pathogens. Th1s can secrete interferon gamma to activate inflammatory macrophages, which are also referred to as classical or M1 macrophages. So these activated macrophages produce inflammatory cytokines and bactericidal molecules like ROS, which makes them better at clearing bacteria. Th1s can also kill chronically infected macrophages through expression of something called Fast ligand, which triggers extrinsic apoptosis, and this removes the bacteria's replicative niche since it's an intracellular bacterium. Th1s also produce molecules that act on cell types other than macrophages. So they secrete IL-2, which is a critical survival cytokine that supports additional Th1 cells as well as cytotoxic CD8s. Th1s can also produce IL-3 and GM-CSF, which stimulate monocyte production from the bone marrow. Th1s can produce inflammatory cytokines like TNF-alpha and lymphotoxin alpha, or LTA, both of which activate the vascular endothelium to promote immune cell recruitment into the inflamed tissue. Lastly, Th1s can produce chemokines like CCL2, which can recruit newly differentiated monocytes from circulation into the infected tissue. And these monocytes, once they enter the tissue, can then differentiate into macrophages. I know that this slide shows a lot of different functions that we're just now learning about Th1 T cells, but what I mostly want to emphasize here is that on top of the innate immune effector functions that we already summarized at the beginning of this lecture, CD4 T cells can act in many ways to further promote innate immune activation and function, whether it's through helping macrophages clear an intracellular bacterial infection, or by promoting the development and recruitment of additional immune cells. Considering what we've recapped so far today, I really want to emphasize that the successful elimination of an infection requires the integration of both innate and adaptive immune responses in order to achieve these first three goals of an immune response. Again, please keep in mind that today's example looked at a bacterial infection in the skin that triggered a generalized type 1 immune response, but the specific cell types and signals required for pathogen clearance will differ based on the type of pathogenic infection and where the infection occurs in the body. After a pathogen has been cleared during this initial phase of infection and inflammation, we've been introduced to this idea that the body then needs to enter the middle portion of this figure shown here in purple, which corresponds to the resolution phase of infection. This stage obviously relates to the fourth goal of a successful immune response, which is to resolve inflammation and return the tissue to homeostasis. And here in this context, homeostasis simply refers to a more balanced physiological state that you would expect to see under baseline conditions. Part of this resolution phase is driven by the death of T and B cells which responded to infection. So again, the activation of these lymphocytes is often accompanied by high levels of cellular proliferation, which we refer to as clonal expansion. However, in the absence of pro-survival signals such as cytokines and antigen detection, many of these lymphocytes will simply die off during a process that we call clonal contraction. However, there are also a number of more active mechanisms of immune suppression. So we're going to end today's lecture by learning a little bit more about some of the cell types and signaling pathways that are important for this resolution phase of an immune response. This is a fairly complicated topic, but for today we're going to simply go over 3 different categories or sources of immune suppression that contribute to the resolution of inflammation. The first is a series of inhibitory receptors expressed on lymphocytes after they've been activated for a while. This figure from the textbook shows several of these receptors, which, rather than containing the activating Itim domains in their cytoplasmic tail, instead have something new, which are called Itim Domains, which stands for immunoreceptor tyrosine-based inhibitory motifs. These are expressed on T cells, B cells, and NK cells, and when they bind to their cognate ligand, they can recruit these ship proteins, which function by removing phosphate groups from molecules that are associated with lymphocyte antigen receptor signaling. So by doing this, this class of receptors basically antagonizes or balances out the activation of signal transduction pathways, which we had previously learned about as being critical for T and B lymphocyte activation. These receptors can also block NK cell degranulation. Second, regulatory T cells are a major source of inhibitory signals involved in shutting down an inflammatory immune response. Tregs can do this in a number of ways, but today we're just going to focus on their production of 2 immunosuppressive or immunomodulatory cytokines. So first off, we have TGF-beta, which inhibits effector CD4 T cell differentiation, and can also function by decreasing CD4 and CD8 T cell proliferation, which again is a key outcome of their activation. TGF-beta can also block the transcriptional expression of cytotoxic molecules like perforin and granzyme by cytotoxic CD8 T cells. Tregs also produce this cytokine called IL-10, which inhibits production of inflammatory cytokines by APCs. Like TGF-beta, IL-10 can also suppress T cell proliferation, and this regulatory cytokine also inhibits expression of MHC and co-stimulatory molecules by APCs. So IL-10 and TGF-beta are powerful immunosuppressive cytokines that can block the activation of both innate and adaptive immune cells following the clearance of an infection. Lastly, the immune system can also play a role in wound healing responses that tend to skew towards more immunosuppressive phenotypes. A good example of this is seen with macrophages. So up until this point in the course, we've primarily learned about inflammatory macrophage activation, which is sometimes referred to as classical activation or an M1 macrophage phenotype. These cells function by presenting antigens, producing inflammatory cytokines, and reactive molecules like nitric oxide and reactive oxygen species that can be used to kill pathogens. In general, M1 macrophages are viewed as a highly inflammatory cell type that can attack invading microorganisms. However, macrophages can come in other forms or differentiation states, including this example of an alternatively activated or M2 macrophage. These macrophages phagocytose debris, but they don't really trigger much inflammatory signaling, so they mostly just break down material and clear it. They also deposit large amounts of collagen and produce some other molecules that are more associated with tissue remodeling and repair. These obviously contribute some important functions to clearing out damaged tissue and debris following inflammatory responses to pathogens, and the function of these macrophages can help bring the tissue back to a state of homeostasis. Note that this presentation of macrophages as M1 versus M2 phenotypes is a little bit of an oversimplification, so I think it's helpful to introduce these 2 different activation states of macrophages on a conceptual level, but keep in mind that in reality, most macrophages probably exist somewhere on a spectrum in between these 2 different extremes. Lastly, as an interesting side note, in these last few weeks of this course, we are going to learn about several mechanisms of immune suppression that are critical for the resolution of inflammation, and that these are often dysregulated in various diseases. These include things like autoimmunity, where immunosuppressive signals don't function correctly, as well as different diseases like cancer, where we kind of have the opposite problem of seeing improper activation of immunosuppressive pathways when we actually want to have immune recognition and killing of tumor cells. We're going to discuss this last point at a much higher level of detail in future lectures, but I wanted to first introduce it here as an upcoming theme in this course. Finally, the last phase of a successful immune response is shown here in green, and this corresponds to the development of long-term antigen-specific immune memory against that same pathogen. In weeks 4 and 5, we learned about some of the pathways that control development of memory T and B lymphocytes, which are maintained for long periods of time. Finally, the last phase of a successful immune response is shown here in green, and this corresponds to the development of long-term immune memory against that same pathogen. In weeks 4 and 5, we learned about the pathways that control development of memory T and B lymphocytes, which are maintained for long periods of time and really accomplish this fifth and final goal of a successful immune response. For the sake of time today, I'm not going to recap memory T and B cell formation, but just keep in mind that stimulating adaptive immune memory is basically the underlying goal of vaccine immunology, which we will learn about in the next lecture for week 8. Again, at this point in the course, you've already learned a lot of the technical details behind these phases and goals of an immune response. I just wanted to make sure that we have a refresher on these topics at a more conceptual level before we start our final few weeks of the class. In summary, today we went over an example of an immune response to a bacterial infection in the skin. We focused on the communication and integration of multiple immune cell types and their functions in the generation of a successful immune response, and we examined how these functions contribute to the overall goals of an immune response as they relate to the clearance of a pathogen, the resolution of inflammation, and development of immune memory. I've highlighted a few of these points below, focusing on the spatiotemporal dynamics of these responses as they relate to early innate immunity, later phase adaptive immunity, and how cells are activated and how they function across different tissues in the body. We've also introduced some of the mechanisms that underlie immunosuppressive signals that are needed to return an infected tissue to homeostasis, and how this is followed by the formation of memory T and B lymphocyte populations. I hope that this lecture has been helpful in recapping some concepts that will be important for the next lecture this week, Lecture 8B, which will cover principles of vaccine design. This vaccine lecture is probably one of the most important lectures for this course from a public health standpoint, so please make sure to give your full attention to the next lecture that's scheduled for this week. You also have an optional Lecture 8C, which will specifically cover the SARS-CoV-2 vaccines that have been developed in the past year.